Sure, if you guys aren't tired of hearing about them, let's talk about the brain organoids, what they are, and how they can be used in the simplest terms. You might be familiar with the term stem cell. That essentially means a cell which can be turned into other types of cells. So, when it comes to the human brain organoids, we take skin cells, which are more similar to pluripotent stem cells. They can't become anything, but they can become a lot of things, and one of those is neurons. This process is essentially the same process of a human being produced in a womb, except we are only growing the brain. As you can see, sometimes they have little eyes. That's the same process that we have when we're being formed. Some of the first researchers who actually made the human brain organoids found that the longer that they grew, the more complex the brainwaves became. And they found it very disturbing, but after about 60 days, the brain organoids seem to have very similar brainwave to a preterm baby. Now, they did state that we don't really have brainwave patterns established for fetuses, for obvious reasons, but we do have them for preterm babies. This has led to a lot of ethical concerns. Some, however, are still pushing ahead to put these little guys on computer chips and essentially immersing them in an artificial world. So all of the information that they get is going to be what we give them. That's why when we made them play Pong, we essentially immersed these little brains into the Matrix. That's all they know. And they are more efficient at learning than AI models because they are actual neural activity, not the versions of neural activity that we make in a computer. These little guys also have great applications for medicine. We can actually look at how medications affect the brain directly. And in terms of personalized medicine, we could make a little brain organoid from you and see how the neurons interact with medications before we give them to you. They also make great models for disease. We could, for example, take the prion diseases, which form plaques in the brain, and give them directly to the brain organoids and see what's actually happening. It's a great way to see what medications might help it too on a small scale. Rather than going through the entire process of working through mice and then humans, we could just go directly to the brain organoids, assuming that it's ethical. And the ethics is a big concern. We don't know what it's like in the little brain organoids' heads. Some say that it's perfectly fine because, they're teeny tiny. How much information could possibly be processed in those little brains? Others say that it's a little bit too close to home. However, we can still use brain organoids from other animals. Mice brain organoids are being used in the US. I hope that helps.